My name is Higgs and this is Boson. And welcome to the Higgs world.
Thanks for the introduction, Higgs. Today we are going to review phylogeny, body morphology, and trophic level shape intestinal traits in coral reef fishes.
In this presentation, our goal is to offer you a quick summary of the research and conclusions. We follow inquiry-based learning model where students take lead in learning. First, they get a quick review of the research via the videos. Second, they review the paper to learn and understand key concepts. Finally, they can follow up with further research on their own to dig deeper. In the end, students would have learned something new, latest and relevant.
Great point, Higgs. Please follow the links to review the research in further details. We want to thank all the authors and publishers for making this research public, thereby advancing science and innovation. Please like, share and subscribe.
Phylogeny, body morphology, and trophic level shape intestinal traits in coral reef fishes. Trait-based approaches are increasingly used to study species assemblages and understand ecosystem functioning. The strength of these approaches lies in the appropriate choice of functional traits that relate to the functions of interest. However, trait-function relationships are often supported by weak empirical evidence.
That is great, Higgs. Processes related to digestion and nutrient assimilation are particularly challenging to integrate into trait-based approaches. In fishes, intestinal length is commonly used to describe these functions. Although there is broad consensus concerning the relationship between fish intestinal length and diet, evolutionary and environmental forces have shaped a diversity of intestinal morphologies that is not captured by length alone.
Very good, Boson. Let me explain more. Coral reefs host an extraordinary diversity of species. Among these species, fishes are the most diverse and prominent vertebrates, exhibiting a wide array of morphologies and trophic strategies. Given this multitude of feeding behaviors, reef fishes represent an ideal group to study how evolutionary and ecological mechanisms influence intestinal morphology. Using a large dataset encompassing 142 species across 31 families collected in French Polynesia, the researchers test how phylogeny, body morphology, and diet relate to three intestinal morphological traits: intestinal length, diameter, and surface area.
That is wonderful, Higgs. The research team demonstrate that phylogeny, body morphology, and trophic level explain most of the interspecific variability in fish intestinal morphology. Despite the high degree of phylogenetic conservatism, taxonomically unrelated herbivorous fishes exhibit similar intestinal morphology due to adaptive convergent evolution. Furthermore, they show that stomachaeless, durophagous species have the widest intestines to compensate for the lack of a stomach and allow passage of relatively large undigested food particles. Isn't it interesting, Higgs?
Very good, Boson. In summary, rather than traditionally applied metrics of intestinal length, intestinal surface area may be the most appropriate trait to characterize intestinal morphology in functional studies. If you like this subject, please follow the links to review the research in further details. We want to thank all the authors and publishers for making this research public, thereby advancing science and innovation. Please like, share and subscribe.